Today I'm going to share a little bit on water and a little bit on not patterns, but I use the word relationships. But before I do that, just a brief background on myself. I was raised in a military family. And we moved around from different locations internationally and nationally and different Air Force bases. Then when I went to college, I wasn't quite sure what to major in. I went to Texas A&M University and goofed off my first year and didn't really have a major and went home for a summer job and thinking I might take a little bit of time off from college, but my parents didn't like that idea, so they encouraged me to continue. So I did a little bit of soul searching and who am I, what's important to me, and I thought, I've always had compassion for people without enough food to eat, compassion for the hungry. So I thought, I'm going to look at agriculture. So I looked in my college catalog, an actual physical book at that time, and saw agriculture. I saw a major called agronomy. Never heard of that word before, but they had introduction to crop science, introduction to soil science. So I enrolled in those classes. I liked the crop science class quite a bit, but I loved the soil science class. We had an excellent teacher, Murray Milford, who helped to show some of that beauty that Dr. Thompson talked about and some of the importance of soils. So I changed my major and have stayed with it ever since then. Just some of the importance of soils, important for plants, water, climate, ecosystem services, pollutants, human health. I have a whole seminar I can give on this slide. I'm not going to do that today, but soils are very important for humanity. For me, instead of patterns, I like to talk about relationships. And for me, one relationship that has been mentioned before on the relationship between faith and science. And I think that God created the earth, so God created the soil, but we humans are charged with taking care of the soil, being good stewards, and serving humanity, and really as an aspect of our faith to be able to take good care of the earth that's provided to us. I was able to write a short article, and I brought a copy along here on the bench. It's called Pray, Listen, and Share Ideas with Others to Enhance Your Soil Physics Research. If you're not in soil physics, you could just say your research, okay? But I wrote it to soil physics audience. Then I think relationships among collaborators. And I've had wonderful collaborators here in the department through my career, outside of the department here in the university, and outside of the university as well. And most of my collaborations have been with graduate students, postdocs, and early career scientists. I was able to write an article titled Set Loving Relationships as Your Highest Priority. This would be in the workplace. And I have a few copies of that up here on the table if you're interested to pick one up. And then finally, looking at relationships among soil properties and soil processes. That's been a lot of the research effort that I've had in my career. So I really can't go forward without mentioning something about my graduate students and postdocs, because they've been such a big part of any science activities that I've had. So a number of my former students and postdocs are faculty. Sang-Ok Chung in South Korea, Jim Hamlet, Penn State, Ibrahim Nassar in Alexandria, Egypt, Gerard Klutenberg in Kansas State, Binayak Mohanty at Texas A&M, Mushtaq Ahmed in Oman, Ming'an Shao in China, Kasuke Noborio in Japan, Tusheng Ren in China, Ibrahim Al-Salama in Saudi Arabia, Daniel Ressler in Susquehanna in Pennsylvania, Jae-Hoon Lee at University of Tennessee, Victor Elia in the Philippines, Frank Casey in North Dakota State, Salam Al-Jabri in Oman, Chanju Wang in China, Tyson Ochsner at Oklahoma State, Josh Heitman, North Carolina State, Dedrick Davis, Alabama A&M, Jacob Prater, and Mike Thompson and I were co-major professors for Jacob. He's in UW at Stevens Point. Aaron Day, University of Nebraska. Yuki Kojima in Japan. Elnaz was postdoc here. She's on our faculty here. And Delia Cole in Israel. In addition to those, I had other students and postdocs that don't have faculty positions. John McBride, Bill Klitich, Masaaki Kiyuchi, Zaki Musli, Jamie Green, Jim Bilski, Toby Ewing, Fulin Shen, Anju Gower, Heath Gieselman, the only one that's a farmer, Xinhua Shao, Sita Ketpratum, Masrur Mahedi, Chenny Luo, Zhuangji Wang, Ohini Akuako, Erica Naidi, and the last student I had, Alam Ramirez Reyes. He's a PhD student somewhere else. Almost all of my work that I've been involved in has been with early career scientists. Some, collaborations with colleagues, and it's just that's been the most fruitful, the most fun, the most interesting part of the job for me as primarily in research, working in the university, is collaborating and working with young people. And then also realizing that there's an impact for multi-generations, because many of those with faculty positions have their own students and so on and so on. I was able to write an article on mentoring. I have a copy here on the bench if anyone's interested, and also have a seminar that I can give on mentoring. Okay, in the presentation today, I'm gonna talk a little bit about some selected research topics that I worked on in my career, and try to briefly explain them, express them, and then I chose these because I think there's more to do, right? So part of this is looking back, but also looking forward. So here's a list: 2D measuring and modeling of soil temperature, water in a row crop, automated tension infiltrometer, a nitrogen fertilizer applicator, heat and mass transfer theory, heat pulse thermal TDR sensors, and some ongoing work on soil structure. Okay, first one is 2D measurement and modeling in a row crop. In this picture, let's see, I don't see my cursor up here, so I don't know what to do, but here I'm trying to show that there's a shadow here in the interrow and a sunlit portion in the interrow. So many times when we do crop modeling a crop, we act as though it's a 1D. That's a uniform everywhere. But we can see from this photo, it's not a 1D situation. It's a minimum of 2D, right? You have perpendicular to the row and you have down. And so when I was in grad school, I worked with one of my fellow students, Oscar Aguirre Luna, and we worked on measuring the temperature distribution in a row crop. Ours was chili pepper in Las Cruces, New Mexico. This is a cotton crop right here. But
that makes me feel better. It looks like cotton to me.
Yeah. Anyway, we worked on chili peppers and we studied temperature. And later, when I was on the young person on the faculty here, young faculty member, I worked on soybean and also did similar work, but included the water. So heat and water transfer in a row crop. So the model we came up with to model the shape of the crop was this hexagonal shape right here. So imagine it's just a rod coming in and out of the screen. That would be the row, and then we would have this row representing two rows. These things representing 2 rows of the crop. What we came up with were the trigonometric functions to show where the sun was positioned in the sky as a function of time and location. So latitude, longitude. We could know for time of year, time of day, where the sun was in the sky. We would know the row orientation. And then we would choose, our height and our width of the row. And then we could calculate as a function of time through the day, which portion of that inner row was in the shadow and which portion was in the sunlight. So we were able to then do energy partitioning. How much radiation is coming? How would that radiation become a net radiation energy? How would that partition into heating the ground heating the air or evaporating water that would be there. So we're able then to calculate the soil temperature and then the water beneath. And we found clearly two-dimensional results. Here's just one slide of information showing the soil temperature. If I stick. Anyway, on the top, very top graph is underneath the plants. You can see there's not much change of temperature. In a relative sense, it's fairly constant. Then the next one down receives morning direct sunlight, so it starts to heat up early in the morning. The third one down is right in the middle, so it receives the most direct sunlight, and that heats up a lot, and even reaches in Las Cruces, it reached 60 degrees C and the surface there. And then finally, later in the afternoon, the other side received the sunlight. So we can see this type of heating and also partitioning of energy, evaporation of water. You know, the microenvironment is quite different. Even a few centimeters apart laterally in space, you can have temperatures 60 degrees to 30 degrees, a big difference. I don't think we have done a lot of work on considering this type of microclimate situation in the soil. I think there's a lot more work to come in the future. Other researchers have used a rectangular shape for the crop, and others have used an oval. So there have been some other work since the time that we did our study, but you know, we published these papers in the early and mid 1980s, and I got shifted onto other things and didn't follow up on this work very much. I kind of shifted my attention, but just about 5 years ago, one of my colleagues from UC Riverside, Jirka Simonek, maybe some of you know his name, approached me and told me that he read these old papers, and then he decided to include this model into this 2D, Hydrus 2D. It's a widely used soil hydrology numerical model that he would put out there. And I was just thrilled that something I worked on basically 40 years ago is kind of getting some new attention. And I think it needs a lot of attention if people want to study, you know, microbial kinds of activities. It's not just a single temperature or water content. It's a whole range of these things, or microclimate energy balance. This type of detail, I think, is important, that is important for us to consider in the future. All right, next topic, tension infiltrometer. I was able to write the backstory. It's in this article up here that I have. If you want to know the backstory on the tension infiltrometer, it's a really cool story. There's not time to really dig into it, but I had a knock on the door of my office in the 1980s, and it was a graduate student. He identified himself as Mark Ankeny. This is a photo, not from then, but a current photo of Mark Ankeny, okay? And so he said to me that he was in crop physiology major here in the department, and he wanted to talk about ways to characterize soil porosity, soil pores, because he was interested in root growing, what kind of soil properties could be correlated to root growth. So we talked about a bunch of things, you know, to look at, but I had in my lab like a homemade tension infiltrometer that we just kind of knocked together. And I said, well, you know, if you can study unsaturated water flow, you know, it's moving through pores, the same pores that roots would grow in. Maybe that would be something to look at. So I quote, loaned him this tension infiltrometer. I didn't know if I'd see him ever again or the infiltrometer ever again. But a couple of months later, he came back to me and said that he redesigned this infiltrometer, and he made a automated. So he put some pressure transducers on. You could hook them to data loggers, and they would automatically measure flow rates from the infiltrometer. Automated infiltrometer that could give very precise information on how unsaturated water flowed in the soil. So I challenged him and said, well, that's great, but you know, how are you going to tie that to root growth? And can you get a soil property like hydraulic conductivity? You have such high quality data. Can you analyze it? Now, Mark Ankeny, maybe had an introduction to soils. I don't think he ever took a soil physics class. He never took my class. He was a crop physiology, very interested in plants, but he is a thinker, and he was thinking and thinking. One day he came back to my office and kind of drew out almost like a cartoon on a piece of paper and said, well, I think if we can measure this and calculate this, then we can, it's almost like one of those cartoons you can see when you're a kid, all these things, and you come up with this answer, big light bulb or whatever. And I was looking at this cartoon. It was just a conceptual thing. And I said, yeah, I think you have something. And so anyway, here's an infiltrometer. Anyway, I knew a little bit of mathematics, and I think mathematics is an important tool for us. I don't know how many of our students study mathematics these days, but a little bit of mathematics is one of those languages and one of those tools that help us to find patterns or relationships. So I solved, a little bit of Richard's equation, transient water flow equation, and came up with these equations right here. They don't mean much to you, but basically what these equations can tell us, if we can measure at least 2 water fluxes at known tensions or matrix potential values, steady water fluxes, then we can determine the saturated hydraulic conductivity and unsaturated hydraulic conductivity relationship at that location. And we didn't really have tools that we could do in the field where we could get the hydraulic conductivity function. So now we did that. ISU patented Dr. Ankeny's device, and it was marketed by a company. And everywhere that I've traveled in the world, I've seen that they have this device. So it was a 20-year patent. The patents run out and everything, but this particular little finding here provided a means to determine the saturated, unsaturated hydraulic conductivity of field soil. And I think this is something important because we have lots of models that we use now, hydrologic models, but they don't have a strong basis on measurements. There may be some pedotransfer functions, maybe try to derive hydraulic properties from soil texture or other things. But in my experience on pedotransfer functions, they need a point of measurement to connect to that space. This is the type of device that can really connect pedotransfer functions to hydraulic properties that will be useful for all kinds of hydrologic calculations that we would do. Secondly, at the same time, we were measuring our soils in Iowa, well-structured, root channels, wormholes, things like that, preferential flow. This is just the bottom of a soil column that's about 40 centimeters tall. And you can see at the bottom one spot, water connected from the surface streaming out the bottom in this one place. You can see a stream of water. We have preferential flow that goes on in our soils here and really all around the earth. But this has a big impact on how chemicals would move in soil, chemicals that are dissolved in the water. Some would move very rapidly if it's in the water, or if it's in the soil, water would bypass those chemicals. So we did some work with Dr. Janes. At the time was in the National Soil Tilth Lab. Now it's National Lab of Ag and Environment. And we figured out how to use a tension infiltrometer, not only to determine the hydraulic properties that I said a moment ago, but we could use some tracers, add some tracers to the water in the tension infiltrometer, and we could be able to determine what's known as the immobile water fraction. How much of the water is readily moving, how much of the pore space is water readily moving, and how much of the pore space is very slow or dead end there. So we had a simple mathematical equation. We would use at least 2 tracers, tracer 1 and tracer 2, as a function of time applied to the soil. And we have a linear relationship where the intercept is this fraction of the water content that's immobile or basically not moving. Then we also, from the slope, we can determine a exchange of chemical between the mobile fraction and the immobile fraction. So we did lots of field work. Frank Casey, others, Jae-Hoon Lee, others, former students did work on this. Anju Gower did work later, Salam Al-Jabri. But I'm just trying to show we did a trend. This is just one transect, but a whole distribution of the immobile water fraction. So based on this model analysis and these measurements, quite a bit of the soil water is not contributing very much to the transport. Only a fraction of that water has fast transport going on in it. All right. So this, I think, is also important because now with this one instrument, we're able to determine not only hydraulic properties, but some chemical transport properties as well. So related to the hydrologic systems of the earth, but also in the sense of distribution of favorable nutrients or pollutants unfavorable, we can obtain some information that are some point source types of information that can be tied to pedotransfer functions. So we can use broader and a wider range of calculations for how water and chemicals move in the soil. Okay, the next topic is called a LCD, nitrogen fertilizer applicator. Has anybody heard of this before in the room? One person, I don't know, online, but we did some work. I did some collaboration with Jim Baker, former from Ag Engineering. I guess it has a timer on it. And Tom Kaspar from NLA and National Lab of Ag and Environment. And the graduate student working on this is Dan Ressler. And this is what our thinking was. For much of the way that nitrogen fertilizer is applied, let me say it like this. If it's applied through a knife, then a piece of metal is put into the ground and pulled through the ground, and it leaves kind of an opening behind it as it deposits a band. So there's an opening in the soil, and then it sort of pushes the soil away from where that knife is. So on the surface, right above that opening is a little depression. So you have a little micro depression, micro topography depression in an open area, and then your band right below that. So in my Horton kind of thinking, I'm saying, if you want to design a system to increase leaching, that's it. You've got a crack, you got an opening, surface runoff will come in here, go right down through the band. So we decided maybe we can change that. Maybe we can close that opening by having a press disc in this case, to compact a little bit right behind the knife, and then have a cover disc to throw some soil from the side so that on the surface there's a little mound. So that's our localized compaction and doming. So it's putting it right above where that knife goes through the field. So in that case, we want the runoff to have water come beside where the band is and infiltrate beside the band rather than through the band. Here's just a shot of the two discs. We did a lot of measurements in labs, simulations, in the field, in lysimeters, and in the field, cornfield. So this is a cartoon, but it shows the data that we collected. This would be a regular knife. You can see this little depression. And after some time, after we fertilize, here's the band location. You can't see it, sorry. Here's the band location in both cases, this little circle. But after some time and rainfall, we had a lot of the nitrogen move down, and there was less in the root zone than with the LCD method. Here's our little mound, here's our little furrow beside. We had much more of the nitrogen remaining up in the root zone in that case with less leaching. When we looked at the yields in a little bit wetter than average yield, we got a little bit of an increase in the corn yield with LCD compared to the knife in a drier than average year, we found no difference. So we didn't see a detrimental effect. People are worried about compaction, right? Nobody wants to do compaction, but this is targeted compaction, and we didn't find any effect on the root growth or the yield from the compaction. So so Iowa State patented this idea and everything, but no company picked it up. I feel like this is a kind of work, nitrogen leaching and that sort of thing hasn't gone away. But this is not one of the methods that I hear talked about as some way to kind of manage that. And I think maybe it still has a little bit of life in it for future investigations. All right, next topic is coupled heat and water transfer. And even broader than that, heat and mass transfer. This, I think, has been, when you're in research, there's certain topics that your heart is just attracted to. This is the topic area that my heart has really been attracted to ever since graduate school and all through the stages of my career as a professor. Thermal gradients in soil, they cause water to move. When water moves, it carries heat. Furthermore, liquid water moves, it carries whatever's dissolved in it, solid. And so coupled together and combined together are the movement of heat, water, and chemicals, particularly dissolved chemicals in the soil. So we did a little experiment. This was an experiment done way back in the late 1980s. Some of you will remember I had a graduate student, Ibrahim Nassar. Ibrahim Nassar did a whole series of measurements where we did a closed soil column. We kept one end at a low temperature, one end at a high temperature, and we had different soils, different soil water contents, different soil solution concentrations, so different chemical concentrations in the soil. From the hot end, we would have evaporation. Water vapor would move to the left and condense on the cold end, and then liquid water would move back. So water vapor is moving this way. It's a dynamic system. Vapor one direction, liquid water back the other direction. And so if it's no salt, we can forget about osmotic driven things. It's just matrix potential gradient bringing the liquid water back. If there's salt present, that has an effect on the evaporation rate. It has an effect on the liquid water coming back. So here's just one figure that we took. It's from an article published in 1989 with Nassar. And it shows one of the results for two curves. One of these curves has salt and one of these curves does not. It's more like distilled water. So these are water content distribution. And this is the hot end on the right and the cold end on the left. Which one has salt? The triangle or the circle? No answer. So the triangle does not have salt and the circle has salt, but thanks for guessing. What happens is when you have salt, then you don't get as much evaporation, right? The osmotic potential doesn't allow the water to evaporate as readily, so it doesn't get as dry. When there's no salt, it'll get drier at that given temperature. And when there is no salt, then that water condenses on the far end for no salt. You have to have a large gradient, large water content gradient, matrix potential gradient to bring the liquid water back to keep evaporating. So it's cycling, right? It's evaporating, condensing, liquid water back, and evaporating like that. But when there is salt, you have less vapor that's going to condense, plus driving the water back, you have the matrix potential and the osmotic potential both driving it back. So you don't have as much of a gradient. When this work was published in 1989, Bill Jury, he's a member of the National Academy of Science, one of the few soil scientists there. He was handling this paper as the associate editor. He told me privately, he said, he thinks this graph, this work, he thinks is going to be a citation classic because he didn't see anybody that had done this or been able to model it, because I'm going to talk about the modeling in just a moment. It's not true though. It's not. It has some citations, but it's not like it didn't go viral, you know, is what I want to say. So sometimes when you're doing work, what I'm trying to say is when you're doing work, you're trying to understand more mechanisms, trying to, you know, be descriptive of those. It takes time for that to catch the attention, you know, in a way that's interesting or usable by a wide variety of people. I think this work is work that, in particular for drier climates, would be important, where you have, you know, salt building up in the soil and salt dynamics like that. And one of my former postdocs, Delia Cole, works in a desert institute at Israel, doing some work on this. And of course, Dr. Nassar from Egypt has done work on this. But I think it's the kind of work that's interesting, but it's really interesting from the mechanisms in all these interactions that are going on simultaneously. So just really briefly, the go-to theory when I was in graduate school, and I think it's the go-to theory right now, is Philip and de Vries theory. They published in 1957. They had more than one article, but coupled heat and water movement in soil. But the assumption that they have in their theory is the water's distilled water. Okay, so that's what we built on. So with the Nassar and these references I have here, we extended, you could say, the Philip and de Vries theory to include the effect of osmotic potential on vapor movement and liquid movement. And we also included the movement of the chemicals itself, right? So we went from heat and water theory to heat, water, and chemical transfer theory. And we did that for both. In the 1997 paper was on inorganic, so dissolved, and the 1999 paper included volatile organics. So this would be chemicals that would partition to the solid phase. They could be sorbed, they could be dissolved in the liquid phase, or they could volatilize in the gas phase. That work in 1999, I always thought that's the most comprehensive theory, but it's not a highly cited article. It is to be discovered and used, I think. But I want to say that a few years ago, again, Dr. Simonek, the chief, the person that's in charge of the HYDRIS modeling, in HYDRIS 1D included the Nassar Horton theory into the HYDRIS 1D. So he did incorporate it into that model. So it is available on the inorganic chemical aspect. So the theory, we won't go into it, but we have a partial differential equation for water, and we'll just say that it has water content change with time, chemical concentration change with time, temperature change with time. So we have these coupled partial differential equations that were solved to describe these processes simultaneously. All right, I have to keep moving. The next one is the thermo TDR, and in the paper up here, I do the backstory on the thermo TDR method. The thermo TDR stands for time domain reflectometry, and thermo is associated with a heat pulse sensor. And so way back when somebody if someone knocks on your door, invite them in, is what I say. So someone knocked on my door and said, you know, they just graduated from Canada. They're in Ames for a while. Their wife is a graduate student in this department in crop science. And he just wanted to know, it's Tu Sheng Ren is his name. He just wanted to know if I could, you know, if I could hire him, if he could work for me. And I said, well, you know, I don't really have postdoc funding right now. So I said, why don't we just start and we'll just, you know, set a time, we'll meet together and brainstorm. And, you know, if we're coming up with some ideas, maybe I can get some funding. So we brainstormed up this idea of the thermo TDR sensor. It's a sensor combining a heat pulse method with a TDR method. Heat pulse method measures thermal properties. The TDR method measures soil electrical properties. and we combine them into one sensor that measures soil thermal properties and soil electrical properties. Here's what a sensor may look like. On the heat pulse, we have a heater wire in the middle rod here, and we can give a pulse of electrical energy to heat. We can measure the temperature increase with time on the outer ones, and then we wire it to a coaxial cable so we can measure the TDR, the electrical signal. So we can do both these measurements. From a heat pulse measurement, all we measure is temperature. We get a temperature increase, a pulse of heat coming by, and we can focus on this one point, the maximum temperature increase. What's the time of the maximum temperature increase? Gerard Klutenberg from Kansas State University, a brilliant mathematician, he derived these analytical solutions to analyze that data so we could determine the thermal diffusivity, the volumetric heat capacity, and from that, the thermal conductivity. So from one little measurement, we can get these three thermal properties. Here's just a graph. You can get the heat capacity, the thermal diffusivity, and I'm not showing the thermal conductivity. Well, over the years, that's 20 years ago, over the years, we've seen we can measure other soil properties that are not commonly measured. And this, I think, to me is the heart of, what I'm excited about and interested in, and I think has usefulness in the future. So in addition to temperature and water content, which lots of people can measure, we can measure the air-field porosity. So all the people that work on greenhouse gases, on movement of gas in soil, it's very much tied to the air-field porosity. So we have ways to measure dynamically in the field air-field porosity. We can measure bulk density, dynamic bulk density. We can put a sensor in the field and do repeated measurements over time to see how bulk density would change. Say for example, you till the field and then the soil settles naturally, or you have traffic on the ground. You can measure the changing bulk density with time. We can measure the thermal properties. And down here on the fluxes, maybe the most exciting in my mind, we can measure the liquid water flux of infiltrating water. If it's raining, whatever, irrigating, water moving down into the soil, how fast is it moving in the soil? Or if water's evaporating from the surface, how fast is water liquid moving up to the surface to evaporate? These are new things. We haven't been able to measure these before. My time is going to run out, so I want to get to the thrust of what we discovered here. Water content, bulk density. These are from the field. We tilled the field, put our sensors in the ground. Then also at different times, we got repeated measurements with the thermal TDR sensors, but we took standard core measurements over time. We found on these two fields after tillage how the bulk density increased and then kind of levels off. These are not the types of measurements that a lot of people do. One of the reasons that they don't do them is we don't have the sensors to do the repeated measurements for bulk density. Bulk density, these are water retention curves. The same soils, if you change the bulk density, you change the water retention curve. If you change the bulk density, you change the hydraulic conductivity. You change the thermal properties. You change the gas diffusivities. All the processes, all the properties and processes would be affected by this bulk density term. But we haven't really measured it because we haven't had a way to measure it. We now have a way to measure it. I think as this gets out, known, this will be a technique that will be widely used in soil science. All right, I really wanted to get to this one. This one I get excited about. This is this is a cartoon, but imagine a soil column that we can control. We can know the flux, the rate that water's moving, the flow rate of water in the soil. So we can control that. We can measure that. Let's just say this is a lab situation. We can control it. We put a heat pulse probe in there, and while the water's flowing, we do a heat pulse measurement. Then we have, from that measurement, we have a a temperature signal that's upstream and a temperature signal that's downstream. So if we first say, well, there's no flow, this is a heat pulse sensor in soil, there's no flow, then the temperature with time curve should be the same on the upstream and the downstream, because it's just conduction heat transfer radially from the heater in the needle. It's going to be the same upstream and downstream. But if there is flow, we have both conduction and convection. So some convection carrying heat downward. So when there's water flow, we expect upstream, the temperature doesn't get as high. And then downstream, because of convection, the temperature goes higher. So we had a visiting scientist from China, Chenju Wang, the same time that a graduate student, Tyson Ochsner, was doing these measurements in the lab. And we were analyzing these. And Chenju Wang looked at a solution to the conduction-convection equation, and he came up with this next relationship. This equation here. This equation here is, to me, a most interesting equation because it's a simple equation. J represents the water flux, how the rate of water flow in soil. And it's proportional to the natural log of the temperature downstream to the temperature upstream. So with a heat pulse sensor, we can measure that temperature downstream and temperature upstream when we give the pulse of heat. And we can also measure the thermal conductivity, that's lambda. Then in the bottom, rho C, that's the heat capacity of the water. That's a known value. And then R is just the spacing between the needles. This is a equation right here. This is an equation that everybody in this room, if you're at all interested in soil water movement, you should learn about this equation. Because to my knowledge, limited knowledge, we do not have other methods that can measure the liquid water flux in soil. Darcy came up with the steady water flow equation in the 1850s. Buckingham, that's for saturated flow. Buckingham in 1907, something like that, had unsaturated steady flow. Richards in the 1900s later, transient water flow. But all of those methods required that you know matrix potential gradient or a water potential gradient and a hydraulic conductivity value. We don't know the hydraulic conductivity values. We can guess them, but they vary orders of magnitude in the soil. This method uses heat transfer to calculate water flow. Convective transfer by water has no hydraulic property, doesn't need a hydraulic conductivity. This is the only method that I know of that can measure the liquid water flow rate in the soil. This is the kind of equation right here that can have a lasting impact on people that want to measure how fast water is moving in the soil. The other ones, you can do calculations, but you do guesstimations on the hydraulic conductivity. This is a direct measurement. I don't know others. So if you don't hear anything else from my talk today, and the beauty of this equation is so simple. It's so simple to use. We have other people, Jan Hopmans, I noticed, was online. He and his group have worked on this particular method, doing improvements on that. And I don't know if Scott Jones, maybe he's online also. He and his group have done work on this equation. They've extended it not only to, this is for 1D flow. They've shown how you can get multi-dimensional kind of flow with the same kind of thinking. Anyway, they can give a seminar later about their work. So here's some results. On the y-axis are the heat pulse estimated water fluxes. On the x-axis, where in the lab, we knew the water flux. We got good relationships between them. Okay, I'm going to run out of time, so I'm just going to say Josh Heitman did a great job using sensible heat balance to be able to measure soil water evaporation. See me for details. We did comparisons in the field situation, a large weighing lysimeter. The weighing lysimeter, change of mass, you can get the evaporation. And we had our little heat pulse sensors in the weighing lysimeter. This was spearheaded by Dr. Wren and his group. And we can see almost a 1-to-1 with the heat pulse. I keep using my cursor, but it doesn't show, sorry. The heat pulse measurements of evaporation and the weighing lysimeter change of mass. We get very good ways. We have very good ways now to measure soil water evaporation, not only cumulative over the day like these, but we can know which depth the water is evaporating. When it's wet on the surface, it evaporates from the surface. As the surface dries, it goes deeper and deeper into the soil, the source of the evaporation. This is a method that can measure that now. Same with freezing. We can determine ice contents. So I have to go to the end because our time's up. So I think in my career working with these people, I did little, they did a lot, but we have methods now to measure transient bulk density, soil water fluxes, soil water evaporation, soil ice contents. The work that we're doing now in collaboration with Josh Heitman and his group, Yangwei Fu and others. Traditionally, we talk about soil structure by looking at the arrangement of the soil solids. Or some people look at pore structure, pore size distribution, and other things. And then they try to use those to calculate a behavior, a property of the soil. It doesn't work well because it's all complicated. What we're doing is we're trying to measure the properties of the soil, like thermal conductivity, hydraulic conductivity, electrical conductivity, those type of things. And then we're going to use those as a proxy to infer the soil structure. We're going to go in the opposite direction. I don't know how it's going to turn out. We're still working on it, but that's my talk. I have a few questions up here, but I think I'll call attention to the bottom one only. Most of my work, or all of the work I've been involved in, we don't do much on biology. Sounds funny, but it's true. We don't account for the root dynamics and their interaction with soil properties. We don't account for the microbial communities and their function. I think the future of soil science related to soil physics, we've done a lot on physical properties and some on chemistry. We work with the soil chemists a lot, but we need to really start working with biologists, crop physiology, whole plant people, and microbiology people, because the whole interaction with the roots and the microbes, to me, is highly unexplored. I think that's our future. Thanks for your attention today. Answer any question that you have.
Let's thank Dr. Horton first. And if you have questions, we understand you may have to leave, but Dr. Horton is here. We should ask him some questions. Great talk.
Yes.
With regard to how you got to where you are today, you talked about when you were first starting in your undergrad, you wishy-washy with it, weren't really sure what you were doing. What was the pathway or transformation like from going from I don't know what I want to do through discovering what you're interested in to the point where you decide to go on to grad school, get a PhD, move into academia instead of maybe industry? How did that all take shape for you?
The question was a little bit more detail on my pathway from undergraduate to being a career scientist. And I think, Mike, my answer is it emerged. It was like an emerging information for me and decision by decision along the way. So when I was a boy, I wanted to play baseball. You know, I was not science. Our family was not on natural resource discussions. We talked about sports and, school, like our schoolwork, but we weren't science. I wasn't science oriented. And somehow when I first took the soil science class, I got interested, and it was more, some people use the word passion, but I just say like an interest. And so I followed it. And then I wasn't sure I would go to grad school. In fact, accidentally I went to grad school because I didn't like my other options, but then I got a taste of research and I liked it. And then I liked soil and water, and then I kept going, but I had tremendous self-doubts if I had the stuff to make it, honestly. But it was more an emerging a series of decisions along the way for me. Thank you for your question. Early in my, I want to fill the gap. Early in my career, when I first got here, Rick Cruz, Mike Thompson, and another colleague, Fred Blackmer, welcomed me here with open arms. And they helped me. They had graduate students. They asked me to, get involved and collaborate. And I'm saying the welcoming community here and getting my career started on a good step was largely due to my colleagues welcoming me here. Yeah.
Well, Dr. Horton, since you mentioned your early career, you replaced Don Kirkham, and some of us here know who Don Kirkham was, and some of us don't.
Yeah.
What was that? What was that like? He was, I think, considered the best soil physicist in the world maybe at that time.
Yeah.
And so yeah.
So the question, I don't know if you're here online, but the question is, what was it like to be hired into the position that Don Kirkham retired from? For me, it was at first scary. I thought he had a big shadow. I mean, physically, he's a big guy. I'm a short guy, and his success and accomplishments were big, and mine were non-existent. Okay, so it was a big shadow there. And I came to work here, and our department head was John Pesic, and I thought I would say hello, that I'm here, and he kind of gave me a little bit of attention from his work, and he said to me, "Okay, well, welcome aboard here. Go and do soil physics, establish a soil physics program." That was the only charge I had from the department head, so it was wide open. It's whatever could emerge would emerge, right? He wasn't giving me any boundaries or charge except go do soil physics. Okay, so I met Dr. Kirkham. I'd met him briefly at some meetings, but not well. And so when he came here, here's what it was like, Lee. I'll just tell it to you straight. We met here. He said to me, welcome. And then he asked me, who did I study with? And I said, I studied with Dr. Van Bavel for my master's degree and Dr. Viringa for my PhD degree. And he said, well, Dr. Van Bavel was my first PhD student. So I'm his grand student. And then he said, Dr. Viringa was a student of one of his students, Don Nielsen. So I'm his great grand student. So we shake hands and it's like, welcome to the family.
Good. Yeah.
And so Dr. Kirkham and I, he worked here for 15 years after that, and we did some collaborations and had many, many good talks together.
That's good. That's what it should be. College should welcome someone. Yeah.
Thank you. Yes.
Would you, I don't know, but would you reflect on your international interactions? You've done a fair amount of traveling, especially in China.
Yeah. So
the role that it's played in your professional development.
So the question is on international collaborations and experiences, and quite a few. It's not like I'm not a jet setter traveling all over the place, but for many, many years, in the summer times, I have done international travels. And I failed to mention on like my list that I was showed of the graduate students and everything, I've hosted about 25 international people, some students and some senior faculty through the years. So it's been quite a bit. I've had international, a lot of work in Europe early. Rink Vanderplug and his group, I would go every year. He would come here. His students would come here. Keith Bristow from Australia, he had a sabbatical here, and I had visits over there. But a lot in Japan and China. It's been more concentrated. And so Ming An Shao, when he left here in the 1990s, he was rising in his career in leadership and accomplishment. He became a lab director and then an institute director like that. And he invited me to be on the International Advisory Committee for these things. And so I would, attend meetings over there and, give some evaluation or guidance like that. And we did research together too. And I had 2 sabbaticals in China. And Tu Sheng Ren, when he left here and he got a post in China Agricultural University, we kept our research very close on the thermal TDR research and Many of his students have been over here visiting. I've been over there a number of times visiting. Very close relationships, I think, in China. I haven't been back since the pandemic though. So 2019 was my last visit, but some Chinese visitors have been here in the meantime. I think it's had a big impact on my career because I found so many students. Again, I love working with students. I found so many eager students in China and Japan in particular that really wanted to talk on science. And I like, as you know me, I like to talk on science and life. You know, why are you doing what you're doing? Have you thought about this? And I like to also at least raise the question of faith and science for people to think about together as well. But I think a tremendous impact enriched me personally and my career as well. Thank you so much for your question. Dr. Thompson.
I really appreciate all of your thoughts about your career, but I want to ask one sort of technical question.
Yeah. Yeah.
And it kind of marries two of the things that you've worked on. I wonder, is it significant? You know, we're targeting placement of fertilizers where the plants are. I wonder if the impact of that on osmotic potential might also have an impact on evaporation of water, whether the water is whether it's evaporated in the row or between the rows. What's the potential connection between osmotic potential and differential?
Yeah, thanks for your question. Dr. Thompson is asking about osmotic potential effects on evaporation, in particular, if there's placement of fertilizers, what kind of effect does that have on osmotic potential and water movement? So let me just say that most of the soil water evaporation, certainly in agricultural fields, occurs near the soil surface. And so if it's a fertilizer that's deposited below the surface, it's not going to have a very big effect on evaporation. So evaporation is, from our measurements here in Iowa, is from the top 5 centimeters of soil is where the evaporation would start from. So not a lot of effect on evaporation per se, but there have been some studies looking at how the fertilizer placement, the osmotic potentials would attract water to it. So it decreases the osmotic potential, and so it decreases the water potential. So higher water potential in surrounding soil would be attracted. Could be some on the liquid phase, but also on the vapor phase. So not so much on the surface balance, but subsurface water movement could be a factor. Thanks for your question.
Excellent. I want to thank Dr. Horton again. Thank you very much, Dr. Horton.
Thank you.